No. Okay. Hi, everyone. Welcome to our first live stream. Hi, Mr. Chow. Hi, David. Okay, so go ahead and ask any questions you want. Otherwise, I guess I'll just random start reviewing stuff. Okay, so while we wait to see if anybody else joins us, let me remind you that your quiz is mostly on pages 61 to 73. So that is signal transduction onward. So signal transduction, cell cycle, mitosis, meiosis, DNA replication. But remember, there will be a also non-table. So that's the transformation practice problems that you can also find on the website if you need the key for that. We did that in class. It's a blue paper. Okay, let's see. So with the all summer none tables, I guess I'll start with that one. Remember that you want to approach it row by row. So you want to think of it as like a, if you have a plasmid, then whatever genes are in that plasmid, you have to take all of them into account, right? So you can assume that if a bacteria gets a plasmid, they're going to get all the genes in that plasmid. So if it's resistant to a certain antibiotic, or if it's the ability to make a certain protein. But that being said, having all the genes doesn't mean you make all the protein. So then you have to read the text in the question, really figure out like what parts are going to be made no matter what. Like if you have the plasmid and you have whatever genes are present there, some of the genes are going to be transcribed no matter what, as long as you get the plasmid. And then other genes, there might be some sort of control mechanism, right? Where like there is a hello. If you have a particular like sugar or something that binds to a repressor protein, then it might stop that protein from being made, or it could be that you have particular protein where it will be made unless something else stops it from being made. So just sort of remember there are those conditions. I would always recommend kind of writing yourself notes as you read those transformation problems. Hi, Caressa. Because if you do, then you don't have to go back and reread all of that text. You should always assume that the media that the E. coli are growing on is plain and it doesn't have all the extra stuff unless you're told it has it. Okay, so let's see, what else would people maybe want to know about? Anyone? Let me take a look at the practice quizzes to see if maybe there's stuff in there that you guys might find important to look at. Let's see. Oh, so something important to know, actually something you don't need to know, I forgot to take it off of the practice quiz. So on one of the practice quizzes, there is stuff about the Schmooing lab. You do not need to know that for your quiz. You should know about signal transduction in general, but you don't need to know about the actual lab itself. So I'm not going to ask you about MAT-A or MAT-alpha or any of those things. What you should know, though, is things like the 3 stages of signal transduction. So reception, transduction, response. You should be able to apply that idea, that concept, to any sort of like example, right? So it doesn't have to be the adrenaline example I gave you in class. It really could be anything that you should be able to, like, if you look at the practice quiz, there is a picture and there's a diagram, and you should be able to look at that and sort of figure out like what are the different parts of the signal transduction pathway in that picture. You should be able to look at a picture and say, okay, what would happen if these things go wrong with it, things like that. You definitely want to be able to do one of the linkage maps. So remember, with a linkage map, you want to make sure that you start with the 2 genes that are going to be the closest together, and those are going to be the ones with the smallest recombination frequency. And then once you figure out those 2, then you add on a 3rd gene, and then the next one, usually there's 4 or 5 at maximum, So in doing so, then the way you'll know if you're right is all the numbers should add up, right? If you look at the data, um, then okay. Okay, so I see a couple questions now. So yes, I will go over it. So in signal transduction, how are chemical signals amplified? The chemical signals are amplified because it's not like a game of telephone, right? Where like one molecule just talks to another molecule and talks to another molecule. It actually gets amplified instead because what you have is you have one molecule binds to, three other molecules, which then triggers like another 10 molecules, right? So every time it advances to the next step um, of signal transduction, like the next reaction, then random fertilization. Okay. I will answer all the questions that are coming up right now. So every time you advance to the next step, then what you end up with is basically more and more molecules being activated by the previous step. So at the end, you could have started with one signal molecule coming in and binding to a receptor, and it could end up triggering like a million molecules in the cell. So that really makes it so that you can have a single molecule have a very large response. Like you don't have to have a lot of signal molecules, which are sometimes called ligands, um, you know, in order to have a response in your cell. Could I please go over the transformation practice problem? I can. Give me a second to pull it up. So while I'm doing that, I am going to temporarily go to Hannah's question. Hi, Zachary. And answer that one, and then I'll come back to the transformation problem once I get it pulled up. Okay. So camp and second messengers, it's just the idea that you have this whole signal transduction pathway, and a lot of them are proteins, and so they're sort of like um, the ones we focus on a lot in the transduction pathways. But in between sometimes those steps, you have these second messengers like camp. And so they're just non protein molecules that are going to serve as like connections in that whole transduction pathway. Sorry, I'm just adjusting my feed here. Neha says, what exactly is random fertilization? I'm not sure what you mean by that. Can you give me some context, like where you saw it? Um, I don't know if you're talking about it in the context of like, um, okay. So yes, and I will talk about the transformation problems right now. So let's go there for a moment. Let me pull it up. If you have it with you, I would say go ahead and take it out too. Um, let's see. Where is my transformation problem? Okay, hold on a second. Let me find a different way of finding it. Sorry, guys. It'll take just a second. Okay, so I'm going to go over the first transformation problem, and then I'll go over the second one, or if you need help with the second one, we can go there too. But I want to make sure we kind of address all the topics too. Okay. So in this transformation problem, again, there's a lot of details that I give you, but you want to kind of be able to pick apart the main stuff, right? Which is to say you have this plasmid. Remember, a plasmid's like a happy meal, meaning if you get the gene, you get the plasmid, you get all the genes. So in this case, you have TET-R, you have GFP, you have BLK3, right? In the explanations, I always tell you a little more about them. So TET-R is going to code for a protein that destroys tetracycline. GFP, we know, goes for the green fluorescent protein. BLK3, now that's the conditional one, right? Well, sort of. That's actually the controller, I guess you could say, right? Because BLK3 codes for a protein that binds to the operon of GFP unless maltose is present. So what I would do here is I would write down, okay, if maltose, right, then yes, GFP, basically, right? If no maltose, no GFP. And in sort of making those notes to yourself, you don't have to go back and read that every time. The rest of the problems, honestly, are always the rest of that portion is usually going to be fairly standard, which is to say you do a transformation. And what you want to remember is transformation is not 100%, which means that you have the plasmids, they're all getting, thrown out there, and they're hoping they're going to go into a particular, into a bacteria, but they might not make it in. And so it's not like every cell gets a plasmid. So at the end of that whole process, then um, then you sorry, I'm just reading the questions, but I'll get to all those in a second. So the idea is that if you then assume, you have to consider every time first, like who's living, who's not living on this plate, and then from there you can continue, right? So what I would really do is, like I said, it's much more practical to look at those transformation problems and go by row, because what you're saying is this is the food they were given and now I'm going to only consider that scenario and then I'm going to move to a different scenario. If you go down by columns, you have to keep shifting in your mind like what's going on. So if you give it plain food, remember LB agar is going to make any bacteria grow. They're pretty much like food, it's like a buffet for bacteria, right? So when I look at the practice problem, I would say, okay, well, oh, and Christine, they are it is a blue one I gave out in class. That's what I'm talking about. If you can't find your actual copy, you can go to the website and you can look at Unit 2. Under Unit 2, it actually says, I think there's a link to the practice problem. So you can print out a blank copy or you can also print out the copy with the answers. Okay, so any colonies means does anything grow? And that means, so if it's LB plain, that means you have to always consider I have untransformed and transformed cells, right? Some that got the plasmid, some that didn't. So the first question is anybody growing, yes or no? Now you could say yes, everything grows. You could say yes, only the ones with plasmid grow, right? So a yes just means something's growing. The key is now for the rest of the row, whether it's 4, 5, 6, however many boxes after that, you're really thinking about then, um, of the ones that are growing, what's true about them, right? So, for example, I would say, okay, for the first one, do any of the cells glow under UV light? Well, they're only going to glow if they're making a GFP. We already established when we looked at the problem earlier, you can only make GFP if you have maltose. So when I look here at the condition, there's no maltose, and you should assume there isn't unless it's stated. So then I would say, well, then the untransformed ones, they can't glow because they can't make GFP to begin with. The transformed ones, they're not going to make GFP because they don't have maltose. So it would be of the ones that are growing, none of them are glowing. Okay, the second one would be, do they produce BLK3 mRNA? Right, again, of the ones that are growing. So since it's LB plain, everybody's growing, I'm considering both the transformed and untransformed cells, right? So the ones that don't have the plasmid don't produce BLK3 mRNA, but the ones that do have the plasmid do make BLK3 mRNA, so that's why it'd be some. On that same note, only some of them contain the plasmid, right? And again, because only the ones that have the plasmid contain the TET-R gene, it's also some. Now, when I move to the next line, it's going to be LB with tetracycline. So this is like food with poison, right? So now I'm saying, okay, I put transformed and untransformed ones on the um, plate, but now all the ones that don't have the plasmid actually don't survive. So for the next 4 boxes in this scenario, I'm only considering the ones with the plasmid. Do they glow under UV light? They still don't. They don't have maltose, so it's still none. But now, since I'm only considering the ones with the plasmid, they all can produce BLK3 mRNA, they all contain the plasmid, and they all contain TED-R. The third row is the interesting one too, because you want to remember antibiotic resistance only comes from some sort of gene like TET-R or AMPR or something like that, right? So they don't naturally have resistance to these antibiotics. So if you now add LB with ampicillin, which is an antibiotic that none of the genes give them resistance to, then pretty much does anything grow? No. And then you can actually leave the rest of the row blank. The fourth one is LB with tetracycline and maltose. So now it's like food plus poison plus maltose. So again, yes, colonies grow, but now only the ones with the plasmid, right? And now because I also have maltose, those ones with the plasmid can make GFP. So it's going to be all of them that can glow. And then the next 3 are also the same as the ones with tetracycline because now they all have the plasmid. So they are making BLK3 mRNA. They do have the plasmid. They do contain TET-R. The last one's also interesting because now you're switching back to where nobody is being killed by anything in the media. So the LB with maltose means it's LB, but you add a little maltose. So now everybody can grow. But on top of that, the ones that have the plasmid now also can glow because they can make GFP. So they're now some of them are going to make BLK3 mRNA, the ones with the plasmid, the one and again, some of them have the plasmid, and they also some of them contain TET-R. Okay, let's see. And so, yes, briefly, the idea of operons is sort of just the idea that you're going to have a group of genes that all get transcribed or translated together. In the context of this quiz for my students, um, again, remember, I don't need you to know anything more than that. Just because there's so much content, I don't want to have it be too little time that you can spend on all those different topics. So this way, just focus on the all summer none table and then 61 to um, in 61 to 73. Okay. Neha, you said notes thing, a collection of genes within one gamete do not give it a better chance of fusing with another gamete. Um, okay. I think what it means in this case, I'm not quite sure where that is in the book, but I think what it means, especially when you say about, talk about in the context of random fertilization, is the idea that you have all these, like just talking about like sperm and egg each have half the DNA, right? And so 23 chromosomes in humans. What that means then is when the sperm are trying to fertilize the egg, it's to a large extent random which one gets in first, right? It's not like there's no relationship per se between the how well a sperm's going to get into the egg and what kind of DNA is in that egg, like which genes it has, right? So I think that's probably what's meant by random fertilization. Do you need to know about CAP and CAMP for the quiz? For me, no. I mean, like I said, just know the all summer none table, and we're good. You will need to remember CAP and CAMP when we get back to our final, but not yet. Let's see. Caressa is asking, how do cyclins and cyclin dependent um, kinases control the cell cycle? I mean, without getting into too much detail, which I don't need you to know too much about, it's the idea that the absence or presence of cyclins along with cyclin dependent kinases is what allows the cell to move from one stage of the cell cycle to the next, right? So if there's something wrong and the cell shouldn't move forward because it needs to correct something, or maybe it's not done replicating its DNA, whatever it might be, then those two like halves of the puzzle won't be there. And then the cell cycle is sort of temporarily stalled until it can do that. And so the levels of cyclins and CDKs, which are cyclin dependent kinases, like vary throughout the cell cycle, and so that's why they being there or not being there kind of allow the cell cycle to continue or not. Madison says, how would you determine which direction DNA replication takes place in? um, well, so the good thing the important thing to remember is that DNA replication can only happen 5 to 3. And what I mean by that is you can only build new DNA from a 5 prime to 3 prime direction. Meaning, sorry, sometimes I try to grab random um, visual aids that I have nearby. Okay, so think of it this way. If ornaments, right, were like the nucleotides. So I'm going to grab some random ornaments from my tree here and hope this works. Let's see. Mr. Chow, can you grab a couple ornaments? Thank you. Ideally, things with hooks would be good. Okay. All right. So our ornaments are going to be our nucleotides, right? And so I have a distinct end to my nucleotides, right? There is the um, 5 prime end, which is going to be this side, and this is going to be the 3 prime end, right? So normally the only way that I can build a nucleotide, or sorry, build a new piece of DNA is by hooking it onto the 3 prime end. That the next one would come in, right? And it would have to hook in like right there, right? And then the next one, sorry, I don't have enough hands to do this, so this is probably going to get a little dicey. Ooh. Okay. Okay. Right. And then the next one come in, right? So I can only build DNA 5 to 3. So in that sense, it has to happen only in one direction. But that being said, now I have to figure out like there's other enzymes involved, right? There's helicase. So helicase is going to start by unzipping the DNA, and so you end up with this fork, right? And you've got these two different sides. One of those pieces of DNA is going to be going 3 to 5 in the direction that helicase is going, right? Which means I can now use my once I do all the other initial parts of building my, my primase and whatnot, then I'm going to be able to have DNA polymerase 3 come in, and just as helicase continues to unzip the DNA, it just keeps following and building a new piece of DNA 5 to 3, and so that strand is going to be my leading strand, right? On the other hand, my other strand is my lagging strand, because that's the one where I can't just follow helicase and keep adding nucleotides on, because it's going to be going in the opposite direction. So what it would do in this case, if this is my strand, right, is it would start to build this way. But it's going to run out of space, and then it's going to fall off, and it has to hop backwards, right? So you're going to end up with two different directions. So I'm not sure if that answers your question about direction. But I guess what I would say, simply put, is you want to look at what direction. I have drawn an arrow, like what direction is helicase going in, and then based upon that, look at the two strands of DNA and say, okay, I have to build something that's 5 to 3. So the strand that allows me to do that in one long strand is going to be the strand that's going from 3 to 5. Okay, so Hannah says, just to clarify, does the DNA polymerase 3 build the new DNA from 5 to 3? Yes. And move on the existing strand from 3 to 5. Well, the existing strand is going to be going 3 to 5, yes, in that direction, right? Since it would be the complement of the new DNA that you build. But remember, there's also all the other pieces to DNA replication. So there's like the primers that have to come on from primase, and then after that you've got, polymerase 3, but then you have to switch out the nucleotides, the RNA nucleotides. So then you would have DNA polymerase 1 coming in, fixing that stuff, and then ligase like sealing everything together. So there's a lot of details, a lot more than like with polymerase, with transcription and translation. Okay, other questions? Anything else you guys have wonder about? Let me see, what else can I remind you of as you're looking for questions? Don't forget, okay. Is restriction enzymes recombinant DNA viruses on the quiz? I don't think that's 61 to 73, right? So I would say no. Unless it's in 63 to 61 to 73, which, as I look at it, I don't believe that's where it is. Let me check. I mean, like I said, it will show up on your final, so it's not like it's never going to show up. It's just, I don't want to make everything, I don't want to overdo it for you all. So, let's see. Let's see. Yeah, because 61 is the beginning of signal transduction. So I'm pretty sure viruses and all that is before that. So no. Know it for the final. So don't completely. Okay, what do SSBs do during DNA replication? SSBs basically help keep the DNA strand open, because remember, helicase is unzipping the DNA, but that's really the only thing it does. So it unzips the DNA and moves forward and unzips more DNA, right? So if once helicase is essentially out of the way, then the two strands of DNA will reform their double helix, and it then sort of makes the whole point of unzipping it sort of not purposeful, right? So what you need to do is actually unzip the DNA, and then you want to keep the DNA open temporarily, so you actually can have all those other players come in. Like you can have your primase come in, and then the replication of the polymerase can come in, etc. So that's what they do. SSB stands for single-stranded binding protein, because they bind to the single strand of DNA and keep it open temporarily so that the actual replication can happen. No, Sophia, you don't need to know scientists for the quiz, so don't worry about those. I gave you those just in the context of so that you would understand sort of how we came to understand that DNA is genetic material, but I'm definitely not going to ask you about those scientists. Okay, Jasmine, for the track transformation practice problem number one, how come in the fourth row all cells can glow? Oh, okay. So the difference between the fourth row and the fifth row, remember, is in the fourth row it was tetracycline and maltose. So all the cells that didn't have the plasmid were already killed off. So really, for the next four boxes, you're only considering the ones that have the plasmid. So that's why it's all of them. In the last row, remember that is no tetracycline, so it's just LB plus maltose. It's like a buffet with maltose added to it. So none of the cells um, were killed. So now in the next four rows, you're actually considering both the transformed, the ones that got the plasmid, and the untransformed ones that didn't happen to get a plasmid. Okay, what else? Let's see. How do protein modifications work in the context of signal transduction pathways? So, one of the ways that, obviously, I don't need you to know like super specific pathways, but if we wanted to sort of be kind of general about, realistically, there are probably some common ways that pathways end up happening. One of the ways that you could have this sort of like domino effect in the transduction pathway would be that, like, it is kind of like dominoes, right? I don't know if you've ever seen like a like a video of like dominoes falling, but like one domino hits the next one, hits the next one, right? So in the context of signal transduction pathways, it could be like molecule, like protein A is activated and then changes shape. And now it goes and binds to protein B, which now changes protein B's shape, which changes protein C. So it kind of creates this cascade all the way down where at the end, it's going to then finally bind to a molecule that's going to trigger some sort of cellular response. So that's sort of the idea of how protein modifications would work in the context of signal transduction pathways. Another possible way that could work too is to say, again, it could also be your final response. So it could mean like all those things happen, and then the final thing that happens in the cellular response due to that initial signal coming in is that a protein maybe that was previously not working now has something bind to it, now it works, or maybe previously it wasn't working, and now it was working and now you bind to it and now it doesn't work anymore. So those are sort of the ways that you could connect the idea of protein modification with signal transduction pathways. Mm, other stuff? Other questions you have? Let's see. When would there be okay, so remember, recombination frequency is essentially it's data that's collected and we're looking at it. So um, high recombination frequency means that more often the two genes, right? The alleles for two genes get separated during crossing over. So, and the idea is that the closer they are to each other, sorry, on the chromosome, the more likely they are to not get separated. Not to say they won't, but their chances of getting separated are lower, right? So you would have a high recombination frequency when you have two genes on a chromosome that are very far apart because their chance of getting separated is now fairly high just because there's so much space in between them, right? Whereas you would expect to see a low recombination frequency if you have two genes that are really close together. Um, the example I gave in class was like, if there was one of those random occurrences where something like split the ground, the chance of you getting separated from the person right next to you is low, and it's a much higher possibility you're going to get separated from someone who's sitting like, across the room from you or even like across campus from you. So that's just the idea. But the idea is that then we can translate that idea of recombination frequency and the number, because if they convert it into a map unit where it correlates to the distance between the chromosomes, then it gives us an idea of how far the chromosomes are physically from each other on the chromosome. Why are there double the numbers of chromosomes in anaphase and telophase? Not sure where you're what numbers you're referring to. Um, in anaphase and telophase, there are not actually there shouldn't be double the numbers, right? Because the idea would be, let me see. Oh, there we go. Okay. So if a snowball, sorry if these are what I have randomly laying around my house. Um, so if a snowball is like a chromosome, right? And one chromosome is represented by one snowball, right? Then before you're going to divide the cell up, you want to make sure that you now have enough for both cells to have a snowball, right? So the first thing would happen during interphase is they would double. And now these are my two sister chromatids. They're identical copies, right? So during prophase and prometaphase, they condense. In metaphase, they would line up. Now, assume the midline in this case goes like across horizontally, right? In anaphase, they would get pulled apart, and then telophase, they continue to get pulled apart. So there shouldn't be double the numbers. No, you could say that early in mitosis and even at stages in meiosis that you have, two copies attached to a centromere or things like that. So you have two sister chromatids and things like that. But remember also the definition of a chromosome changes. So using humans as an example, right? You start the cell with 46 chromosomes, you copy those chromosomes, but now every chromosome is made up of two sister chromosomes. So I still have only 46 chromosomes. And then when I divide it out in mitosis, I still have 46 chromosomes because after mitosis, now I again define each chromosome as one single copy. I hope that, Hannah, I'm not sure if that answers your question, but let me know if there's something more specific or if I can clarify. Anything else? I shouldn't say anything else, but what else? Let's see. I'll look at my transaction pathway is phosphorylation. Like one molecule goes and adds a phosphate to the next one, which activates it, and now that goes to the next molecule and phosphorylates it. Um, so that's a phosphorylation cascade. It's just that it's each one phosphorates the next one. And in doing so, it's activating it, right? Because remember, when you bind something like a phosphate to a protein, you change its shape. And if you change its shape, you may then also then activate it, where before it wasn't working, now it is. Okay, for the lagging strand, just to clarify, does DNA polymerase 3, while synthesizing Okazaki fragments, follow DNA polymerase 1? Well, really DNA polymerase 3 would be going first, right? Because DNA polymerase 3 is the one that's making the fragments. So what you're more likely to see is DNA polymerase. Okay, I think I see what you're saying. Like visually, it might look like it's following it, right? Because it's like hopping backwards and building the strand. So it's not that I guess I would just be careful to say that it's following polymerase 1. They are going in the same direction, though, yes, because both are only able to synthesize DNA in a 5 to 3 prime direction. So visually, it might look like polymerase 3 is following polymerase 1, but really, it's more like polymerase 3 is like farther up, closer to where helicase is, probably making the newest strand of DNA, while polymerase 1 is going to be sort of a little farther down that chain because what it's doing is actually going in and replacing the primer that was used to get DNA polymerase 3 started, right? So it's going to look like it's following it, but it's actually not. Um, Hopefully that kind of clarifies it. Let's see. What else can we talk about? Feel free to type things. But I will take a look at the practice quiz again and see. Do make sure you understand sort of also, the idea behind diploid and haploid. Um, how those processes are different too for like mitosis and meiosis. Um, oh, okay. Why are so many cells in a okay, so think of it as like your cells only divide when they need to, right? If your cells just divided all the time and um they never like, they didn't have a reason to, then you would basically be like growing at a ridiculously high rate, right? And so, and yes, I will get to allosteric regulation in a minute. Um, so the idea is that for a lot of your cells, like just doing their normal cell thing every day is being in G0 because they don't have a need to divide. Cells will typically divide for reasons like either like they have received a growth signal, like a signal transduction pathway has been activated, it's telling them to grow and therefore make more cells, or it could be that there's been damage to cells because um, of an injury. So it's saying now, like, I need you to make some new cells, right? But it's that's not the case for a lot of cells. So they're not getting that message to divide a lot. So that's why a lot of cells are in the G0 state because they don't have a need at that moment to divide. Let's see. How does signal transduction cause changes in the cell? So remember, there's all these different proteins in the cell, but sometimes they haven't been triggered to start working. And so sometimes what causes a process to happen in a cell is going to be internal. It could be certain molecules that inside are triggering different reactions to occur. But it's also possible, which is going to be in the case of signal transduction, for a external molecule, right, like a hormone, to come and bind to a cell, and in doing so, activate this pathway that now sort of gives the pathway that green light to say, okay, now I need to make this particular protein or I need to open this particular channel or things like that. So signal transduction would cause changes in the cell because if that signal triggers a certain pathway that then produces certain proteins that then cause um, certain things to happen in the cell, then it would change the cell. Okay, so re-explaining allosteric regulation. So remember, allosteric regulation is one of the things we talked about in the context of enzymes and how enzymes will one way of controlling an enzyme. And so for allosteric regulation, it's literally the idea that you have, first of all, it's a non-competitive form of regulation. We tend to talk about it, the inhibition portion, but it doesn't have to be. So with allosteric regulation, what you have is you have this molecule that's flipping between an active and an inactive state, right? And in doing that, you you could either lock it into the active state or the inactive state. So the way I kind of think of it is like if someone is flipping a light switch, like on and off and on and off and on and off, right? So an allosteric activator would be something that locks the light switch or the molecule in this case, right? Into an on or active state, and now it's going to be working all the time. Whereas an allosteric inhibitor would be one that locks it into the inactive state and now it sort of prevents it from working. Yes, the difference between DNA polymerase 3 and polymerase 1. So they're both polymerases in that they are both building DNA. They both can only build 5 prime to 3 prime, and they can all they both need some sort of hook to sort of get things started. The difference becomes in sort of what it is that they're building. So polymerase 3 is your main building enzyme. It's the one that's really constructing most of your DNA, right? But remember, in order to build that DNA, you needed that primer hook. And the primer in our cells happens to be made out of RNA. So if you really want to copy all of your DNA, you then at the end have to go in and like cut the RNA out and replace it with DNA. So polymerase 1 is the one that actually replaces it, um, replaces the RNA primer now with DNA after it's sort of gotten things started. Number 9 in the 2017 practice quiz. Okay, let me take a look at that and see which one that is. Is it the 2017 or the 2007? Emily, are you referring to the one that has to do with yeast schmooing? If that's the one you're talking about, then no, we don't need to do that. I think at the very beginning I did mention it, but I'll mention it again. On your quiz, you do not need to know the schmooing lab. Okay, so Cressa says, how do second messengers play a role in signal transduction? Remember again, they're just part of that like domino effect. So it would be if I equate it to the dominoes, it would be like if most of the pathways are proteins, right? But on occasion you have a molecule that connects like one protein to the next, but it's not a protein, like it could be something like camp, then that would be like a second messenger. So if I go with my domino analogy, it would be like if in this whole like domino chain, you have like one intermediate where like a domino hits a marble, where the marble then goes and hits another domino, right? Then the marble is like the second messenger because it's a non protein. Can I explain polyploidy versus aneuploidy? Yes. In both cases, it deals with the results of having too much DNA or not enough DNA. So, um, oh, okay, that number nine. Okay, so give me a second and I'll go to that one. Polyploidy and aneuploidy um, have to do with having too much DNA as a result of non-disjunction, most likely, right? So polyploidy is a little more specific. Polyploidy means you have too much DNA, meaning you're supposed to have two sets of a chromosome. And because of non-disjunction, you have three usually. Aneuploidy is a little more generic, right? Because aneuploidy just means you have an abnormal number of chromosomes. So in this case, now, if you have an abnormal number of chromosomes and it's not, it could be too many or too few, then that's what aneuploidy sort of entails. So again, you can kind of think of as polyploidy is a type of aneuploidy. Okay, so the number nine that has to do with the Stefan regulation of gene, that one I believe has to do with like transcription factors, right, or something like that. Let me check it out just for a moment. So let me make sure that that's what we're talking about. Oh, I see it. Okay, the 2017 one. Okay. So no, for the Stefan one, this one has a lot to do with transcription factors, which is in that portion of your notes that I said we're just not going to worry about right now. So no, you don't need to know that one for this quiz. Let's see. Okay. Um, yes. So the lysogenic cycle is one of the two ways that a bacteriophage could basically reproduce itself inside the bacteria. It is much more covert, right? So the lytic cycle is like it goes in, it takes over the cell, it uses the cell's machinery, it makes a bunch of new viruses, and then it leaves, bursts out. Lysogenic cycle, like I said, I think of it like as an undercover spy, right? Where it goes in and it becomes part of the DNA of the bacteria. So now every time the bacteria divides, you now have a copy of your virus in there too. Until it now from that switches to the lytic cycle, it'll basically be slowly and quietly amassing this army of infected cells. What is a retrovirus and what role does it play in the immune system? So a retrovirus is a virus, first of all, so it isn't necessarily playing a role in your immune system. I mean, your immune system is fighting against it. Um, a retrovirus means that it is an RNA virus. So at the core, the nucleic acid is a RNA instead of a DNA. Now, what that means though is now there's like an extra little issue, right? Because if the virus itself is non-living, um, then it's going to have to get into the cell, take over it, and make that cell make a bunch of copies of virus. And so normally, if it was DNA, it would just say, okay, here's the DNA. Now it's going to use the DNA to make copies of the RNA and the RNA to make copies of the protein, right? And it's also going to be able to take its DNA and just make copies of the DNA to make more nucleic acid for the virus. When we get to a retrovirus, then it's different right now because what's happening is you actually need this um, RNA to be able to go into a cell and use the RNA to make more RNA. But if you think about it, there is no process built into a cell that allows it to make more RNA from RNA. So the retrovirus has to take with it the what we call the reverse transcriptase, where it's going to take in an enzyme that's going to allow it to make RNA into DNA, and now it can now fully take advantage of the cell's machinery because now it has all the pieces to do that, where it can take the DNA now and make a bunch of RNA. In terms of the role it plays in your immune system, again, it doesn't necessarily um, play a role in the immune system, but it's become one of the things that's more challenging for scientists to fight against because your immune system generally tries to recognize parts of a virus and then activate your immune system to get rid of those pieces. And so the problem with the retrovirus is because of that reverse transcriptase, it's a very sloppy enzyme. It makes a lot of mistakes, namely mutations. So when those mutations occur, it now would mean that your every time it goes through the process, it would actually like look a little different. And now it means that um, your immune system won't recognize it, right? Because it tricks it, basically looks different. Um, where do protein modifications fit into the stages of um, signal transduction? Again, protein modifications is one way that the signal transduction pathways can happen. Where sometimes the idea is in order for like step A to lead to step B to lead to step C, where it could be that the first protein is binds something and now it's modified, it's changed, right? And now that allows it to bind to the next protein, which allows it to bind to the next one. So a lot of times protein modifications is how you sort of go from um, reaction to reaction in the signal transduction pathways. PDGF and why it's needed during cell cycle. So PDGF was also just an example of how you might have molecules that would trigger things to happen in the cell cycle. So PDGF is typically only released when you have an injury to an area, right? And the idea is if you have an injury, that cells have died, and if cells have died, you need to replace them. So with PDGF, the idea is that your platelets, which are also in your blood, would release PDGF, and that basically signals to your cells to start dividing, so that you can make new cells to replace the ones that may have died. So that's sort of the idea behind like what it is and why it's needed. It would be needed essentially to sort of kickstart the cell cycle and get things dividing, since it means you need more new cells. Okay. Anything else? Um, Apoptosis and necrosis. Okay. So basically, the difference is let's start with apoptosis. So with apoptosis, what happens is it's programmed cell death. And so what that means is when your cells get to a certain point, a lot of times it has to do with how many cell divisions they've made, and so therefore um, how many how short their telomeres are, then it's going to um, trigger those cells to die. And so and that's a very normal part of your sort of cell maintenance, right? That when cells get to a certain point, they essentially are triggered to self-destruct. Necrosis is more the idea, and my students, we didn't go here, so don't worry about it. Is like necrosis is the idea like when probably because of some sort of very specific event, then like all or most of the cells in a particular tissue or organ are dying. So it's just it's a little more dramatic, I guess you could say, right? It's very targeted. It's usually the result of some sort of thing like an injury or disease or something like that. Whereas apoptosis is more a very sort of regular, almost like maintenance portion. I'll say, for your cells. Um, oops, wrong window. Um, yes. So just so you know, this live stream, what will happen is once we end it, um, YouTube, you have to give it like 15 minutes, I think. But basically, YouTube will automatically convert it to a video, and then you can just watch it as you would any other video from here. So don't worry. I think it does take a little bit of time though. So like once we end the stream in about like 10 minutes or so, then just give it a little bit of time. Usually when I come back and check to make sure it's uploaded, it's usually there after like 20, 30 minutes. Um, how can homologous chromosomes contain the same information but not be identical to information to other? So this kind of gets at genetics, right? This is actually a really good connection to our genetics, even though we didn't talk about it this way. Um, remember that you get each half of your homologous chromosomes from each of your parents, right? So that means like your parents each gave you a chromosome 1, a chromosome 2, a chromosome 3. And let's just say, and I'm going to make this up because it's not true. Let's say chromosome 1 has on it the gene for whether or not you can roll your tongue into a little taco, right? Like that.
Mhm.
Um, oh, thank you, Mr. Chow. Yes, Mr. Chow can do it too. Um, so that being said, it could mean that for your two copies in the homologous pair, right? Let's say you're heterozygous for tongue rolling. So that means your mom gave you the tongue rolling allele, but your dad gave you the non tongue rolling allele. Then that would make them. That was what I mean. That's what I mean when I say it's the same information. Like they both have a tongue rolling allele, but they're not identical because one of them has the non tongue rolling allele, and one of them has the tongue rolling allele. Erica says, how do we know what the different steps of transaction will be for the different cells? You don't. I mean, I don't expect you to know that. I mean, for me, I would want you to be able to look at a diagram or look at it or read about an example and kind of figure out which portion is reception, what portion it would be transduction, and which one would be the response. That's really the level I need you to know that. I mean, I certainly don't need you to memorize any sort of pathway. For my students, no, you do not need to know plasmid mapping for this quiz. You don't even need to know restriction enzymes. Um, can I explain protein modifications for transaction pathway? Yes. Again, the idea is that sometimes, like, think of it in other ways. Like before the pathway is activated, right? You have all these molecules that are sort of ready to be a part of the pathway, and so sometimes the way that that sort of gets going is you have to activate those proteins. And so the way you activate them would be by modifying them, right? Let's say you add a phosphate to it, or you add another molecule, right? And so the idea is that as you now add a molecule and it binds to the first molecule, then it binds to the second one, then that molecule, hits the second molecule, which activates that. And so that's what I mean when we say like protein modifications, where you can like successively down the line, you keep like activating each protein after another. And in doing so, it's going to sort of make the signal get passed down. Can I briefly explain meiosis? Yes, I can. So remember, meiosis, first of all, compared to mitosis, is very different because the end goal, the end product, is a haploid cell. So really, you have to take your diploid DNA, your 46 chromosomes, and you have to find a way to divide the DNA into these four cells that are each going to have half the DNA you started from. So what you're also going to have then is two rounds of division. You are going to have interphase at the beginning where it's still going to copy, right? But now what you have is, and so you still do have at the beginning of meiosis 46 chromosomes, um, but and each chromosome is made up of two sister chromatids. The major difference comes in during meiosis one, because in meiosis one is the major stage at which you're going to now make that cell haploid, right? Because you're going to take the two homologous pairs, the two chromosome ones, and this is a major difference from mitosis. You're going to line them up double file. So it's like square dancing, right? Where the chromosomes, the two number ones line up, the two number twos line up, the number threes all the way down. And then during anaphase one, they separate, right? And then telophase, you form the new cells. So now you have these two cells that each have one of each of the pair, right? So one number two, one number three, number four, etc. All the way down to 23. And the other one also has half. Um, and so at that point, the cell is haploid, but you actually can't stop yet because even though you have only one copy of each chromosome, remember we did interphase at the beginning. And so with interphase at the beginning, now you have two copies of each chromosome. So even though number one that's in the cell, even though it's only half of the pair, is still made up two sister chromatids. So then in meiosis, you're going to go through another round of division where it's basically just like mitosis, and you now split the sister chromatids. So at the end, If you compare those cells to the cell you started with the very beginning, it is half the DNA than you started with. Yes. So the errors in crossing over that can happen basically happen because if the cells are not lined up. Let me see if I can find something that would be. Let's see. See if anything on my Christmas tree works. No. Okay. So think of it. I know it's gonna be a little hard to visualize this, right? But if you think about crossing over as the two um, the two homologous chromosomes are going to switch pieces, right? So if you and your friend are standing right next to each other and you are going to do something akin to crossing over, then it means you would be like switching parts of your clothing, right? So like you're switching shoes, for example, or you're switching hats, right? Um, and so in order for that to work, you have to do it where they're lined up with each other, right? Like the two shoes are at the same level, the two hats are the same level. So um, in an error in crossing over, one of the things could happen, right, would be if they're offset. If like one is higher than the other, then when the pieces break off and they switch, then they're not actually switching equivalent amounts. So that would mean, like, let's say you are standing on a stool and your friend is on the floor, right? And you're supposed to switch whatever's like, 5 inches from the floor, then you don't give your friend your shoe, but they give you their shoe. And now your friend has no shoes and you have two shoes, right? So that's one of the things that can happen where if you now compare you and your friend, right, there's two different errors that have happened. You have like an insertion, you have like too much stuff, and they have a deletion. They're missing something. They don't have any shoes. Another thing can happen is what we call an inversion, right? Which is where the pieces break off, but they somehow get flipped upside down. So that would be like if in the process of taking your shoe off, your shoe gets flipped upside down and now you're wearing a shoe that's upside down. Another thing that can happen is what we call translocation, which is it breaks off and it's supposed to reattach to the other, homologous chromosome, but it doesn't. It gets attached to something totally different. So that'd be like you take your shoe if they take their shoe off, and then some of your shoe goes flying over and lands on like a totally different pairs, um, you know, desk and such. So hopefully that helps. Where are the checkpoints? The checkpoints are present at various stages in the cell cycle. They tend to be present sort of at major junctions. So, for example, like making sure that DNA has been copied, right? So there's, I think, a checkpoint between like the S phase to G2. I think there's also a checkpoint at some point where it's like G2 to mitosis, right? So the idea is you want to have these stoplights where like if things are not good, you can slow the cell down. And if they are okay, then you allow it to proceed. Um, how do cyclins and cyclin dependent kinases work? I mean, for our purposes, you just need to know that they are molecules that when bind in combination, right, they kind of are those sort of the puzzle piece that allows you to move forward in the cell cycle. Do you have two pairs of homologous chromosomes in both meiosis and mitosis? You do. So you have pairs of homologous chromosomes at the beginning of meiosis, right? But by the end of meiosis one, the pairs are not present anymore because they've been separated. For mitosis, they're actually there the whole time. But that's the interesting thing too. If you think about it, like in meiosis, the pairs matter, right? In mitosis, they really don't matter. Like even though you have two number ones and two number twos and two number threes and two number fours and two number fives, etc., they really never get together with their pair. They're functioning as one single chromosome. They've copied themselves. There's two sister chromatids, and they're going to split in half. Um, so in doing that, you're not going to now. Yes, the chromosome pairs are there, but it's almost like it doesn't matter that they're there because they never got together. They never paired up. There was never any um, process or reason for them to get together. Okay. Any last questions? Looks like we're almost out of time here. So if anyone has any last questions, we'll go ahead and take those. Otherwise, we will wrap it up shortly. Okay. What are some Oh, okay. So, what are examples of external and internal signals that trigger signal transmission pathways? So, any pathway being triggered, first of all, has to be triggered by some sort of molecule, right? So, an external signal would be, for example, like a hormone that is coming from somewhere else in the body, and it serves as that molecule that's going to start the reception stage of signal transduction. So, an external signal would be like it binds to that receptor and now causes that to start the transaction, reactions, etc., and ultimately insulin response. You could though also have like an internal signal, meaning you could have the cell make a molecule, right, would then bind to a receptor, maybe located just inside the cell membrane, and then it would, you know, same thing though, cause this whole process to happen. The last problem set for B. Um, I can. The idea there is just that remember again that you have two ends to the um, DNA, right? So I'm going to use my ornament here, right? 5 prime, 3 prime. Oh, that was, yeah. And the idea then is normally what I would do is I would hook it onto here, right? And normally, but if there's something blocking it, um, like a card, right? If now there's something blocking and I can't do that, then I would have to polymerize from the other end. So the idea is that in that particular problem, right? The DNA, it's not that the DNA runs backward, it's the idea that if you're going to use this particular nucleotide, since you cannot add a new nucleotide onto that end, then you would have to have it, but you can on the other end, then it automatically would make sense that it polymerizes or builds the DNA now instead of one direction in the other direction. Let's see. Does a virus count as an external signal for signal transduction? Typically, no. I mean, a virus typically is going to bind to receptors, but then the idea is it binds to a receptor sometimes and then it gets taken in. It's not that it necessarily triggers a pathway in the cell. I mean, at least not generally speaking, right? So I would say, generally, I would say no. Okay, any other questions? No? Okay, so I guess unless anybody has any more questions, I'm going to start wrapping this up. Um, just a couple quick reminders, and you can always come in. I guess we don't have tutorial tomorrow, huh? You can come in tomorrow to see me if you have questions during brunch. Um, I do have a meeting during lunch, so I won't be there during lunch. But you're welcome to email me if you have questions as well. And just let me know what other questions you have. And it's getting late, so you guys should go to sleep or get some rest. Because early morning tomorrow, right? Okay. Oh, how long are Okazaki fragments? That's a good question. I'm actually not sure how long they are. I mean, I don't think they're super short. Um, I would guess somewhere in the order of like 50 to 100 base pairs maybe, but that's totally a guess. The idea is just is that they are fragments, right? That they can't make the whole strand all at once. So you want to make, that's why it has to be constructed in pieces though, right? It's because you build a little bit and then you build a little more. But don't worry about how long they are. Actually, I couldn't tell you. I'm not sure. Okay, I guess we'll wrap it up now. Like I said, if you come up with more questions, please feel free to let me know. And you're very welcome. Okay, have a good night, guys. I will see some of you tomorrow. And, well, I'll see any of you tomorrow that care to come and say hi. Okay? Oh, you're very welcome. Okay, guys, have a good night. You're very welcome. De nada. Good night, everyone. Bye bye. Oh, Mr. Chow says, but good night. Oh, here. Oh, good night. Okay, bye bye. Oh, Madison says, "Hey, ciao, wow, wow." All right.
Is that Maddie?
Yes, I think so. All right. I'll see you guys later. Good night.